Welcome to Yaka Digital Network. We often credit soil quality and weather for a successful harvest, but beneath the topsoil, agriculture is driven by invisible biological engines. Maximizing crop and livestock yield requires a precise understanding of the mechanical and chemical processes occurring inside individual plant and animal cells. We will deconstruct this cellular machinery across three pillars: how nutrients cross cellular boundaries, how enzymes catalyze biochemical metabolism, and how DNA dictates the genetic blueprint. This is Senior 5 Agriculture, Cellular Biology and Genetics. Mastering these first principles provides the only systematic method for agriculturists to engineer precise solutions against drought, disease, and consistently low yields. Animal cells are enclosed by a flexible membrane, allowing for varied shapes and movement. Plant cells, conversely, require strict structural rigidity. This rigidity comes from a tough outer cell wall, working in tandem with chloroplasts, the organelles responsible for photosynthesis. These specific structures determine how a crop physically stands, grows, and produces energy. But for these structures to function, nutrients must cross the cellular boundaries. This relies on three specific transport mechanisms. Diffusion is the passive movement of particles from high to low concentration. Osmosis targets water molecules crossing these boundaries to balance solutes. Active transport occurs when the cell expends energy to force nutrients backward against their density gradient. These microscopic rules create macro-level farm problems. High soil salinity creates an external solute concentration that physically reverses osmosis, rapidly starving crops of hydration. Once nutrients cross the cell membrane, they must be processed. This requires biochemical catalysts known as enzymes. Enzymes dictate the speed of metabolism. They break down complex proteins and carbohydrates during livestock digestion and manage energy consumption within crops. Their efficiency is extreme. The enzyme catalase, for example, violently breaks down toxic hydrogen peroxide into harmless water and oxygen in a matter of seconds. To measure this metabolic efficiency, we apply the scientific method. We begin by setting an aim and hypothesis to test the effects of the enzyme amylase on starch. The procedure requires a chemical indicator. We use iodine, which reacts specifically with starch molecules. Here we see three laboratory beakers. Adding iodine instantly turns a solution blue-black if starch is present. The analysis confirms enzymatic activity. As amylase breaks the starch into simple sugars, the blue-black indicator color disappears entirely. When agriculturalists optimize the temperature and pH conditions for these specific enzymatic reactions, they directly accelerate livestock growth rates and crop maturation timelines. Enzymes perform the chemical work, but they are built according to a strict genetic blueprint: DNA. DNA is a physical biochemical compound. It can be isolated and extracted using basic materials found on a farm or in a classroom. The mechanical extraction begins by mashing a common fruit, like a strawberry or banana, physically breaking the rigid cell walls we identified earlier. Once the mixture is filtered, we introduce cold alcohol. This chemical addition alters the solubility of the solution, forcing the DNA to precipitate. The extracted DNA separates from the cellular waste, becoming a thick, white, stringy substance visible to the naked eye. Proving that DNA is a tangible, manipulatable substance allows agriculturalists to treat inheritance and physical traits as precise variables they can control. To read and manipulate that blueprint, we use Mendelian genetics, a predictive mathematical tool. Consider a community that requires a maize variant possessing two distinct traits: high grain yield and high drought resistance. We cross a low-yield, highly drought-resistant parent with a high-yield, drought-susceptible parent. This Punnett square maps out these traits, separating dominant and recessive alleles. The first combination shows how dominant traits mask recessive ones. We then calculate the remaining probabilities to reveal the full statistical distribution for the future generation. Interpreting these results gives us the precise mathematical probability of producing an individual offspring that successfully inherited both target traits. Applying Mendelian genetics removes the guesswork from farming. Communities can systematically calculate breeding patterns to build biological resilience against climate change. The predictive models in the laboratory dictate the practical reality of managing an active farm. Through project-based learning, these biological first principles must be taken into the field to accurately evaluate and improve real crop health in the community. Mastering cellular transport, optimizing enzymatic reactions, and mathematically predicting genetics are the required mechanisms for maximizing agricultural production. Master the micro, optimize the macro. You can watch more videos for all classes here and get all content at www.yucca.cc.